In this 3-mark subjective question, they are directing us to specify the role of the high temperature primer and Thermophilus aquaticus enzyme in which way it is used in the polymerization chain reaction technique. For this, we are bringing the CBSE centric PCR picture for better narration, but in the exam not necessary to draw. In this picture, we can see when we are exposing the double-stranded DNA to high temperature or denaturation, the double-stranded DNA is getting unwinded to single-stranded DNA. To this, we are making the primer to get attached at the 3' end, followed by the DNA polymerase enzyme application. This enzyme is taken exclusively from Thermophilus aquaticus bacteria. Why? Because it can sustain at high temperature. So that is why we are making the dotted DNA stand by using the Pantel stand. In this way, we are representing the PCR to satisfy the given specification. Again back, we are illustrating the illustrated message sequentially to get the respective 3-mark for the first optional question. In the first optional question, they are directing us to specify why we need high temperature. Why? Because to bring the denaturation or the double-stranded DNA. For this specification, we are getting 1 mark. Why we need the primer? Why? Because we need the primer to facilitate the polymerization process. For this illustration, we are getting 1 mark. Why we need Thermophilus aquaticus? Why? Because this enzyme taken from this bacteria is heat tolerant. It is in a position to do the DNA synthesis process. For this specification, we are getting another 1 mark. In this way, we are satisfying the overall 3-mark demand of the first optional subjective question. In the second optional subjective question, they are directing us to specify why we need to choose beta galactosidase coding sequence as a selectable marker. Why it is a better choice than the antibiotic resistant gene. For this, we need to give the appropriate illustration. For this, we are specifying when a recombinant DNA is inserted within the coding sequence of the beta galactosidase enzyme making gene, it results to the insertional inactivation phenomena, so that the enzyme won't be formed. This criteria we are keeping as a selectable marker criteria. For this illustration, we are getting 1 mark. And we need to specify the chromogenic substrate gives blue color colonies. If the plasmid does not have an insert, it is representing the non-recombinant. In the presence of insert, no color is produced. So that is representing recombinant. In this way, if you are illustrating, we will be getting another 1 mark. And for specifying the selection of the recombinant due to the inactivation of the antibiotic resistant gene is a cumbersome procedure. Why? Because it requires a simultaneous plating having different antibiotic. For this specification, we are getting another 1 mark. In this way, we are satisfying the overall 3-mark demand of the second optional subjective question.